So I am joined today by Dr. David Huss, who's VP and Head of Research of Shape Therapeutics. Shape is a relatively new biotech company, and I'm going to let Dr. Huss explain what they do. First of all, thank you so much for joining, Dr. Huss. I know you're really busy, and so this is wonderful that the community gets a chance to get to know you and get to know your company and the program that you're pursuing for Rett syndrome. And before we get into the meat of the presentation, I'd love to just have the community get to know you a little bit. And so maybe we'll start off, if you could just tell us a little bit about your background.
Yeah, certainly. And let me just start by saying thank you so much for the invitation. It's really an honor to be here and to be able to talk with the patient and parent community in Rett. So thank you for that opportunity. My background, I grew up in the Philadelphia area. I would say that I was exposed to science at a very young age. I remember, so my dad is a chemist, and so I remember him bringing home some fun things from the lab where we would do experiments in the kitchen. Everything from that to getting your bug collecting kit where you're out in the backyard pulling, catching butterflies and then looking at them under a microscope. And so in general, I've been very interested in science from a very, very young age. And that led to pursuing a degree in biology in undergrad. And then I went on to grad school to study molecular, cellular, and developmental biology, where I was really exposed to a great mix of a lab that had a head, Dr. Amy Lovett-Racke, who is a PhD immunologist, and then Dr. Michael Racke, who was a MD neurologist. And so it was a really great mix of basic science, but also applied to very specific clinical diseases. And so I think at that point, it became quite evident to me that I wanted to take all the basic science training and really apply it to specific human health problems. And so from my time in graduate school, I then left and joined a biotech company on the East Coast, really focused on trying to understand how the immune system and immune responses lead to some of the problems in multiple sclerosis. And so I would say that I'm a card-carrying immunologist, but I've always been in and around the brain with a lot of the diseases that I've focused on working on. And so from my time working in multiple sclerosis, I became very interested in T-cell immunology and how T-cells interact with different proteins inside the central nervous system. But from there, I moved on to a company out on the West Coast where I was very focused on, again, studying T-cells and how we could modulate them using different molecular technologies that had really been discovered, things like the CRISPR-Cas9 system, where you could now do precise editing in a cell in a culture dish and really try to understand how we could change those T cells to then inject them back into patients to fight different types of cancers. And so again, that was where I was really kind of exposed to many of the molecular technologies that the field had been building. And so from there, Francois Vigneault, one of the CEO and co-founder of Shape Therapeutics, he and I had met, and when he started Shape, he tapped me on the shoulder and said, there's this new molecular technology. It was built in Dr. Prashant Mali's lab down at UCSD. And let's think about how we could build a company around this and what diseases would make the most sense to apply these novel new molecular technologies to. And so that's really what led me to Shape, just an excitement around what we are now able to do at the molecular level, but still the long path it takes to go from that initial idea to actually being able to treat patients. And so as a company, we're really building all the pieces to enable us to move these early technologies towards clinical development.
Great, thank you. That was really helpful. So just to give our audience a little bit of perspective. So as many of you know, RSRT is focused on strategies that attack the root cause of the disease, Mecp2 mutations. We think that by doing so, we'll have the greatest opportunity to improve symptoms. And we're pursuing strategies in 6 different areas. So gene therapy, DNA editing, RNA modifications, which include RNA editing and what Shape is doing, as well as RNA transplicing, Mecp2 reactivation, and protein replacement. And so what we're going to be talking today about is RNA modifications. And so if you are ready to share your screen, we'll start with the presentation.
All right, certainly. All right. Are you able to see the full screen of the slide?
Yes, we are.
Okay. So today I'm really going to talk about the core technology platforms at Shape Therapeutics and how we're really focused on this concept of using systems that are already present in your cells as a way to do gene therapy and gene correction. So many of the approaches that you see in the field right now are really focused on the delivery of foreign proteins for gene editing purposes. And so we've really built a technology platform that is based on harnessing natural systems in your body. You know, to start, I thought it might be.
David, let me just, when I think of questions that the community might have, I'm just going to jump in if you don't mind.
Certainly, yep.
So that's a really good point that you made, right? To harness the body's own natural, phenomenons that are already happening in the body to leverage them for what you're trying to do. Can you just maybe mention, what are the advantages of that?
Certainly. So, as an immunologist, the first thing that I think about is with any new therapy is how will the immune system respond to this? Our immune systems are incredibly good at recognizing what is a natural, normal protein in your body and what is a foreign protein that's been delivered. This is the reason why we mount immune responses to bacteria and viruses. And so when you look at some of the treatments that are used, and I think that particularly around the CRISPR-Cas9 system, which has incredibly valuable opportunity, particularly for doing out of the body editing of cells. And then as those cells divide and they're reintroduced, you do not introduce those bacterial components back into the individual. But one of our fundamental concerns is that when you're expressing one of these foreign proteins all over your body, that you will mount a very potent immune response to these. And so when we were forming Shape and really thinking about an ability to do things like gene editing or RNA editing, but being able to harness proteins or components that are already inside your cell, so your body is not going to mount an immune response to these because it does not, it sees them as natural and not foreign. And so that really for us was one of the key approaches, was this ability to recruit proteins or components that are already present in the cell.
Yeah, no, that makes a lot of sense. And from RSRT's viewpoint and the community's viewpoint, I think the more shots on goal, the better. So the more avenues we can cover, the better it is. Okay, thanks.
Yes, and I think to that point, we talk a lot about there's no one tool that's going to solve every problem. And I think, we cheer on all of the other approaches that are going after some of these very challenging diseases, because I think the more people you have thinking about how to solve problems and the more shots on goal you take and approaches, the more likely we are to find ultimately meaningful treatments. And so I thought it would be worth just starting with a little bit of a narrative on Shape Therapeutics as a company. I would say that we've been able to assemble a team of what I call very passionate problem solvers. These are people that have backgrounds in molecular biology, neuroscience, biochemistry, that have really come together with a single goal in mind, and that is to discover meaningful treatments for some of the world's most daunting genetic diseases. And so I think at the core of it, we're problem solvers, and that's what we really love to do. The reason that I'm talking with this group today is because Rett syndrome is one of the key areas where we are applying this problem solving, really focused on taking these disruptive gene therapy technologies. And when I say disruptive, what I mean is that ways to really have complete shifts in the way that we treat diseases, and focused on applying these to Rett syndrome. So that's really what I'm going to focus on today. When we think about building a successful therapy, there are a number of different elements that come together. The first, you have to have a safe and effective, what I call payload technology. And this is the molecular tool that you're using inside the cell. you have to have a safe delivery technology. And this is really focused on targeting the right cells. And then lastly, and particularly important in the gene therapy space, is an ability to manufacture your drug product with high quality and high consistency. At Shape Therapeutics, we have teams across all three of these different areas where we took a step back and looked at the landscape of what had been done in gene therapy to date and really identified a number of key challenges that are still limiting the field. And that's, again, that concept of identifying problems and then really putting teams of smart people in them to try to solve those problems. And so today, what I'm going to focus on is two of our technologies. The first is a payload technology that we call RNA Skip, and I'll talk in more detail about that. And then the second is our AAV ID delivery technology that enables us to identify AAV capsids to more effectively deliver our payloads to the cells of interest. So if we think now that it's been over 20 years since that initial discovery where it was identified that loss of function mutations in a gene called Mecp2 were the causative genetic factor in Rett syndrome. And this was really monumental for the field because, as Monica stated, we now know what that causative factor is. And so it has enabled researchers to really focus on, at that root cause, how can we make corrections? However, there are still a number of challenges that make drug discovery in Rett syndrome quite difficult. The first is something called cellular mosaicism. So in each female, every cell has two different X chromosomes. The Mecp2 gene is on the X chromosome. And through a process called X chromosome inactivation, in any given cell, you will have one of those X chromosomes silenced. And so what this means is that in a patient, you have a certain percentage of the cells that will have a wild type or a natural copy of Mecp2, and then you'll have a certain percentage of cells that are expressing a mutated copy of Mecp2. And there are some documented studies showing that the level of X chromosome inactivation and skewing can also alter the severity of the disease. But what this means from a therapeutic standpoint is we really want to focus on targeting the right cells with therapeutic intervention. So only having activity in the cells that have that mutant copy of Mecp2 while leaving the healthy cells untouched. One of the other challenges in drug discovery for Rett syndrome is that the level of Mecp2 expression must be very tightly controlled. So we know that loss of function mutations in Mecp2 lead to Rett syndrome, so where you have a lack of Mecp2 expression. But we also know from other studies that if you have an overexpression of Mecp2, so if you overshoot the expression level, this can also have negative consequences. And so any therapy really needs to try to restore the right level of Mecp2 to enable therapeutic benefit. So with these challenges in mind, I think the key question that many in the field have asked themselves is how do we create a targeted therapy that will only have activity in the mutant cells and also restores the appropriate level of Mecp2? And so this is really what we're going to focus on today. At Shape, we have a number of different technology platforms that we're developing to solve some of these key challenges in Rett syndrome. Our goal is really to be able to develop treatments for every single Rett syndrome patient. We know that there's often not a one-size-fits-all, particularly when we're talking about genetic diseases. And so we have a number of different technology platforms that we think will be able to advance not only our own payload technologies, but also other companies through our novel delivery engineering. The first platform that I'm going to talk about is something that we call RNA Skip. And this can be applied to nonsense mutations, where we're able to engineer tRNAs that are able to read through nonsense mutations. And I'll talk more about that in a few slides. We also have an AAV ID platform that enables us to engineer AAV capsids with superior central nervous system targeting and optimal safety profiles. And this is how we think about the delivery of novel payloads. And then the last technology that we are pursuing for Rett syndrome is very, very early, And I won't go into any details about it, but it's really meant to be able to treat patients that have mutations that are not nonsense mutations. So to be able to capture the other 70% of patients with different types of mutations. I want to pause and just take a moment to, number one, thank the Rett Syndrome Research Trust. We've had a very productive partnership with them over the last 2 years. One of the things that has really impressed me about RSRT is the dedication to providing researchers with different types of tools that really help advance Rett research. And so two in particular, one is the access to cell lines that enable us to test our therapies in culture, and then also to be able to help with access to different mouse models where we're able to test experimental therapeutics in some of the early preclinical testing. And so I really think that not just for Shape, but for all of the different researchers that RSRT makes these resources available to, it's incredibly beneficial to advancing our understanding of how to treat Rett syndrome.
Thank you for that. And I'm just gonna, turn that around and thank the families, many of whom are on the call today, because it's their fundraising efforts that makes it possible for us to develop the resources, which then companies and scientists can use. So it's partnerships all the way around, families with RSRT with scientists and companies and academics. So.
All right, so now we're going to dive a little bit more into the science of what we're doing. And so I'm calling this Molecular Biology 101 because I think the start here is really to set the stage for what I'll talk about with our targeted therapy approaches. So if we think about how you have normal DNA in a cell, and so you have this path where DNA is then transcribed into RNA, which is displayed in the middle of this slide, where you go to a single stranded, and each of these little blocks would be a different letter in the RNA code. And then this is read and translated into the functional protein. And these proteins are really what are essential for the function of the cell. When we think about different types of genetic mutations, these largely fall into two primary categories. The first is what we would call a premature stop codon or a nonsense mutation. This is where a 3-letter code no longer tells the protein machinery which amino acid to insert, which is what these little circles on the chain is, but instead it says to stop the translation process. And so in the DNA, this mutation is then also found in the RNA. And then what happens is you get a truncated protein, which oftentimes will go for degradation.
And just to know, so these are all the mutations that end in X. So 255X, 168X, 270X, just so everybody's clear.
Correct. A second type of common mutation would be a point mutation that doesn't lead to a stop codon, but instead leads to a change in the code of that mRNA that then leads to a mutated protein, which could be dysfunctional, have less function, or even have toxic activity. And so if we think about this path from DNA to RNA to protein, there are many companies, and in fact, a lot of the biopharmaceutical industry spent many years developing things like small molecule inhibitors or antibodies to target at the protein level, but that really did nothing to the underlying genetic code. We have had the emergence of a number of different technologies that are able to change the genetic code itself. And while I think there is some upside potential to these, they also do have the risk that any type of off-target change could be permanent and has a level of associated risk with that. One of the reasons that Shape has really focused on targeting this intermediate, what we call the messenger RNA, is that it enables you to make precise modifications that, number one, do not carry that risk of permanent DNA damage, but then number two, they are very tunable in nature. And so if we think about this as a way to turn up the volume or turn down the volume rather than an on or an off. And so the majority of our technologies are really focused on ways to modify this middle bucket of RNA. The technology that I'm really going to focus on today is one that we call RNA Skip. So when we think about the DNA that then goes to RNA, there is a protein complex called the ribosome, which is shown here in kind of this blob in the middle, that is what I call the reader. So it reads along the mRNA, and for each 3 nucleotides that it reads, there is a corresponding transfer RNA or tRNA, which is depicted here, that matches up with that 3-letter code. And each of these transfer RNAs carries a specific type of amino acid. And when these come together, you have this growing chain that at the end of the messenger RNA transcript, you have this ribosome falls off and you have this fully functional protein that will be made. Now, in the context of a nonsense mutation or a premature stop codon, what happens is that ribosome is reading along, but then it encounters one of the codes that says this is the end of a chain, the ribosome should fall off, and you should stop with protein production. In a nonsense mutation situation, you have that happen, and then you don't get that full-length protein made, so you don't have a functional copy. In this context, it would be Mecp2. But what we've developed at Shape Therapeutics is a version of one of these tRNAs that now is able to slot in and recognize the specific nonsense mutations that are found in Rett syndrome. And so what happens is as the ribosome is moving along, instead of having this gap where then it falls off, now we're supplying an engineered version of one of these tRNAs that the ribosome doesn't know any different. Our tRNA suppressor comes in, and then that ribosome can keep reading, and you see a full-length protein produced. And so this is what we call RNA skip, because we're essentially skipping through that nonsense mutation. As Monica mentioned, there are a number of different nonsense mutations that are found in Rett syndrome. So we know that a specific one that is where you have the arginine amino acid and that this is mutated to a stop codon. This makes up around 90% of all nonsense mutations and 30% of the total Rett mutation burden. And so the very common ones are the R168X, R255X, R270X, and R294X. In our labs, what we've done is we've taken these tRNAs that we have engineered, and we've tested those in neurons that we've harvested from mice that have specifically the R255X mutation is the data that I'm showing you here. And so along the bottom here, you have 9 different candidates that we have tested. And then on the y-axis here, we're looking at the percent of Mecp2 that we're able to restore. So in these mice, you detect very little Mecp2 expression without any type of manipulation. And then when we add in these different engineered tRNA candidates, what we're able to see is that you get a restoration of Mecp2 expression. And with some of our candidates, we're seeing a very high level of restoration. Now, I want to be careful to say that this is very early work. So this is still at the experimental phase where we're testing these in mouse models. But we are now moving these candidates into testing in cell culture models from neurons that have been derived from human pluripotent stem cells that were provided from patients that have specific Mecp2 mutations. And so we'll be able to really be able to understand how much Mecp2 restoration are we able to provide. One of the other things that I failed to mention of the importance of targeting RNA is that because we are only going to be able to take the natural level of Mecp2 expression, the RNA that's produced is really what caps that. We can only restore up to 100% of Mecp2 expression without overshooting it with our approach. So with these engineered tRNAs, the way that we deliver those as drug products is using AAV as a delivery vehicle. Now, AAV stands for adeno-associated virus. This has been really phenomenal for the field of gene therapy because what it enables you to do is to use a virus that is able to target different cell populations, but there are no known diseases that this virus causes. And so it enables us to essentially hijack it so that we can use it to deliver our therapeutics. And by using AAV, what happens is you have this AAV will infect a cell. It takes and deposits a small piece of DNA. And with this piece of DNA, what we do is we engineer it to encode these different RNA skip molecules. And so inside the cell, you almost have a little factory for our drug product. And so this is then constantly producing these engineered tRNAs so that in a neuron, for instance, once you have infected with this AAV, you have this DNA episome that is there, and then you have a constant supply of these RNA skip molecules to help correct and produce full-length Mecp2 protein. The other important thing is that this enables you to use a single one-time treatment because this piece of DNA stays within the cell for the life of that cell. And because neurons are the primary target population in Rett syndrome, neurons, once established, you have very little turnover or very little cell division. And so you're able to have a long-term treatment with a single administration. Now, while AAVs have been quite successful as delivery vehicles, there are a number of challenges and limitations in their broad use. And that is why we've really built the second technology platform that I'm going to talk about today, which is our AAV ID platform. And that is really geared towards engineering these AAV capsids so that they have superior ability to target the central nervous system, particularly neurons, and also improved safety profiles. Now, there are a number of products that are on the market for other diseases that are using AAV as their delivery mechanism. And so we know that these can be delivered safely and effectively. However, one of the things that we know about AAV is that in the natural state, they have a very high prevalence of targeting the liver. And so what that then leads to is you get large amounts that are found in the liver, but you don't have as much that is able to get into other tissues, for example, the central nervous system. And what that causes then is you have to increase your doses quite a bit so that you're able to put more and more towards the central nervous system, but that really overburdens the liver in a patient. And so there's a huge need in the field to develop capsids that have better targeting profiles. And that is what we've really been focused on. And I will mention that while this would be quite beneficial to deliver the payload technologies that we're developing at Shape, this is also something that would be beneficial for the field in general, as others are developing other technologies, but they could be able to package those with our novel AAVs. So how do we tackle this challenge? The way that I think about this is what you're trying to look for is an AAV that on the surface is able to have a different type of display that will interact with the target cells that we're trying to get to. And at Shape, we've really assembled a team of scientists that have built careers over building things like high throughput screening platforms. So instead of testing one thing at a time or 10 or 100 things at a time, we're able to build platforms that we're testing millions or even billions of unique sequences at a time. And so what this enables you to do is to test many, many, many more variants all in the same experiment so that your likelihood of finding one that works goes significantly up. Our AAV ID capsid discovery platform is built upon starting with up to 100 billion unique variants. And so this really maximizes our chances of finding the right capsid that we're looking for. These are then passed through a step where we make sure that they're still able to assemble into a productive virus. And so this we normally see in the single digit billions, and so up to 3 billion unique variants. This is then injected as a pool into a non-human primate, where we then let the animal tell us which capsids get into which tissues. And so the way this is done is after 4 weeks, we are able to harvest different tissue types. One of them that we have focused on is the central nervous system. And then we have a way to harvest out and to identify which capsids were able to infect which tissue types. And so the idea is that if you were to give an IV injection of an AAV, it has a novel capsid surface that enables it to really focus on targeting neurons. And then you're able to find these sequences and then be able to repurpose them now and combine them with novel payload technologies. And there's a lot of bioinformatics that is done because you can probably imagine that by starting with such massive numbers, you have to have a way to filter through the information and to identify the individual capsids that have properties that you're looking to select for. So in our first campaign that we've done in an attempt to find capsids that are able to target the central nervous system, we started with 1.7 billion unique variants. And what I'm showing you here in the middle is a graph that looks at, in the different colors in each vertical bar, these are the 20 different amino acids. And so that is the sequence variation. And then in this first campaign, we've targeted 9 different amino acids that we know are important for driving that effect of binding to target cells. And so we're trying to vary these so that they now, instead of infecting a hepatocyte in the liver, they will infect a neuron in the central nervous system. And so the data in the middle shows that we're able to build this library and maintain very high levels of diversity. So there's a lot of uniqueness that's going into this experiment. We then go through that AAV production step. So again, this removes any of the variants that do not productively make virus. And then you have 1.7 billion unique AAV sequences that go into the non-human primates. Each of these 1.7 billion is represented at between 10 and 50,000 copies. And so what we're able to ask is, in a specific site, do we see 10,000 copies of the same variant? And that would tell us that it's very highly tropic, or it very effectively infects that tissue.
David, you're delivering these AAV IDs without a payload. They're empty?
So yes, what they do though is they have a code that when we recover them out, we can tell what was the original starting sequence of that capsid.
Yep.
But it's a good question because the follow-up experiments, after we identify ones that have the profile of targeting neurons in the central nervous system, then what we will do is combine them with our novel payloads. So as I mentioned before, really after we are able to harvest these tissues from the non-human primates, we move these into an entire back-end bioinformatic analysis. And the goal is to be able to have a catalog of these AAV ID variants that have very specific infective properties. And so in the context of Rett, we're very interested in targeting the central nervous system. There are some other diseases where targeting cardiac tissue would be optimal, or having a better way to target the lung epithelial cells would be optimal. And so we're in the process right now of taking all of the sequence information that we've been able to, and all of the capsids that we've been able to recover out of these different tissues, and we're moving that into a process of validation where we'll be able to filter and narrow down and select variants that have the optimal properties of interest. And so I'll say stay tuned, because this work is ongoing right now.
Yeah, it's so impressive. It's a lot of heavy lifting here.
Yes, and I will say, I have the advantage of putting a lot of this into just a few slides. The scientists on the team that are on the ground really developing and cranking in the labs on this, a single slide could be 1.5 years of someone's work. And so I think it's just the reality that, technology development and drug discovery is very, very hard, and it takes time. But I think it's clear that they're problems that are worth solving, because it can have a meaningful impact in people's lives. And so I've talked about the RNA Skip and the AAV ID technologies. Again, I'm not going to go into any detail on our RNA switch technology, other than to say that it's really, we think, an early novel way that we can treat a wider array of the mutation types in Rett syndrome. And I really want to close with just reiterating that as a company, we are very committed to solving some of the drug discovery challenges in Rett syndrome. And we really appreciate our partnership with the Rett Syndrome Research Trust. We appreciate a lot of the doors that has opened for us to interact with other researchers in the field. And while as a company, we are relatively young in our pursuit, we have a lot of excitement in what we're doing. And so I think that over the coming years, it will be very, very interesting to see where we're able to take these technologies and hopefully have an impact when we move these towards the clinic.
Great, thank you very much. So I'm gonna take some questions now. So here's a good question from Eric. He says, is the mRNA toolkit discussed here similar to that used by the mRNA COVID-19 vaccines? Does the massive recent investment associated with these vaccines benefit the work that you are doing?
So that is an excellent question. The short answer is that there is no direct correlation with what we are doing, but I think more broadly in the gene therapy space, there will be a lot of learnings that come from that. And so to be clear, what the COVID vaccination, the mRNA vaccine does, and what others in the gene therapy space are trying to do as well, is by delivering a new mRNA into the cell and then allowing the cellular machinery to translate that into protein. It's used as a method to try to, in the case of a vaccine, it is displaying a portion of the COVID protein so that your immune system is trained to mount a response. In the context of a rare genetic disease, it would be a way of delivering that mRNA intermediate to then have transcription and production of the full-length protein off of that. At Shape, we're very much focused on taking the RNA that's already present in your cell and that's very well regulated off of that Mecp2 DNA and making the corrections there.
Okay, great. Someone else asks, I'm not sure how to pronounce the name, Chiron. Would this technology also apply to deleted genes? I think they mean deletions within a gene, like a deletion of 10 base pairs or something, instead of mutated genes.
So the RNA Skip technology that I talked about would not. That is very specific for nonsense mutations. Our RNA switch technology that we are developing, we hope will be able to address both missense point mutations, but also deletions, insertions, frame shifts, different types of mutations as well.
Okay. Also, I'm getting a lot of questions about, will the webinar be recorded? Yes, we are recording and we will make it available. Probably not until tomorrow, but because we want to add a transcript so then it can be translated in different languages. So stay tuned. We'll have it in a day or two. Okay, here's another question from Thibault. I hope I'm pronouncing that okay. Could someone, this is a good question, could someone that previously had a gene replacement therapy benefit from your treatment? So I think, if you've had AAV previously, would you be able to have it again? That's really the question, I think.
Yeah, so that is a great question. And one of the big challenges with using AAVs is that once your immune system has been exposed to it the first time, you're still able to have an effective transduction of cells that first time, so the payload can get delivered. But subsequent redosing is currently not able to be done with the same AAV type. And so one of the things that at Shape we've also focused on in our first AAV ID campaign that we talked about is using AAV5 as the starting place and then mutating off of that. The reason we chose AAV5 is because it is the most distinct from other common AAVs. And so if you compare the sequences to things like AAV9, which is another popular AAV for targeting the central nervous system, they are distinct enough that they have a predominantly non-overlapping immune response. And so one of the goals is that as we identified variants with novel tropism, we would also be able to evade any type of pre-existing immunity. The other important thing is that when you look at natural exposure to AAV5, it is quite low in the general population. And so that was another reason why AAV5 was chosen as our starting place. But all of that to say that the specific case and individuals' specific exposures to other AAVs will ultimately determine what the immune profile looks like and whether they will be amenable to treatment with any future AAV gene therapy.
Also, there are companies that are working on trying to make AAVs kind of go to like stealth mode.
Yes.
So they would stay under the radar of your body's immune system. And, you know, I think the issue of being able to reuse AAV in gene therapies is a problem that everyone, you know, that lots of people may face. And so there are efforts in the industry to try to solve that problem. And I expect it will be solved. You know,
Absolutely. And I think one of the other avenues that's being explored right now is not altering the actual AAV capsid itself, but thinking about, are there ways that we can essentially trick the immune system so that it doesn't know it's being infected with an AAV? And so there are approaches where people are trying to either modify B cell responses or T cell responses, different key elements that are responsible for viral surveillance, so that you could open up a window in the individual where the immune system would not recognize an AAV. And so those are other approaches that are being tried right now as well. Because as Monica stated, it is a general challenge across the field.
Yeah. So I'm getting a lot of questions about timeline. When do you think this might be in a clinical trial? And I know that that's a tough question to answer, and you want to be careful about managing expectations. But you know, maybe you can take a shot at a projection, you know, which we understand could change.
Yeah, so maybe I'll preface this by a couple of different things. One, you know, at Shape, we really pride ourselves on being science driven and really letting the data guide our decision making. And so because of that, I would say the phase that we're in is in some of the animal testing. That is a phase that you naturally will do to precede any type of clinical trial. And so we would really like to let that data drive the decision making of how fast or how soon we can move something forward. And so I'm not going to put any type of timeline on it today, because I also don't want to provide any false sense of hope. We really want to make sure that we do the work and then let the data drive the decision making of how fast things can move forward.
Yeah.
So I apologize, that's a bit of a non-answer.
No, I understand. I think I want to make the point that Rett syndrome is Shape's lead program. So I think as parents, we can feel comforted with the fact that they're going to put a lot of effort behind it because it's their lead program. So they're going to work as fast as they can possibly work. And, you know, within 6 months, a year, whenever there will be updates, and we'll bring those updates to the community. And so obviously there's questions around missense mutations as well. And I know you don't want to go into much detail, but is there anything else that you can add in terms of, you know, because I'm sure 70% of the people on the call today have children with missense mutations. And so,
right.
Yeah.
I think maybe if I share a little bit about even how we really focused in our energies on Rett syndrome. You know, it's a strange place to be, where you have this really neat molecular technology capability, but you're trying to think about what diseases make the most sense to apply it to. And we initially started looking at Rett because there was such a significant portion of the population that has these nonsense mutations. But once we started investing and building out all of our internal knowledge, hiring in neuroscientists and really interacting with different thought leaders in the community, we recognized that only having a therapy that would treat 30% of the population for us wasn't good enough. And so that's where we really started to advance our AAV ID to be a way that we could use as a delivery tool, not only our payloads, but also others. And then the RNA switch technology, which I apologize that I can't go into more detail on, but that is a way that we are trying to target the other types of mutations, whether they be missense mutations, frame shifts, deletions, et cetera. And so the best I can say is stay tuned. We hope to be able to talk about that more in the future. This also goes along with the theme of it's an early technology, and we would like to let the data drive our ability to move that forward, and really make sure that we're focusing in on solving those core problems and then making it publicly available once we've been able to do a bit more work.
Okay. Fair enough. I'm also getting a lot of questions about age ranges of children or adults that would end up being in a clinical trial. And I think that's probably a premature question to be asking Dr. Huss at this moment. You know, some of the gene therapy trials, the Novartis one, maybe the Taysha one, which probably will be in the clinic, not probably, will be in the clinic before Shapes, maybe, you know, they'll give us maybe some insight. And you know, the reversal experiments that were done by Adrian Bird suggest to us that, you know, because Rett syndrome is not degenerative and brain cells aren't dying, and because the reversal of symptoms was so dramatic in the mouse, even though there is a big leap from mice to people, but still it suggests to us that we should be able to see improvements in older adults as well. I think a full-blown cure is probably asking for a lot, but I think dramatic improvements is probably within the realm of possibilities. So we don't know yet, you know, whether the FDA would say you've got to go in young children or adults, or but my hope, certainly, I have a daughter who's 24, so obviously my hope is that there will be improvements seen in adult patients as well. Let me just look through. Do you have anything to add?
Yeah, the one thing I would add to that is, you know, I think the reality of running those early stage clinical trials is that you try to have as narrow of a patient population as possible so that you can really see if there's a meaningful benefit to the treatment. And then you can expand that out to younger, older patients from there. And so I think that is not just specific to Rett, that's really in any different type of drug development. You really want to start with that first patient population of being very narrowly focused so that you can really understand how your treatment works in a very controlled setting. And so I imagine that it will be quite similar with the first studies for any therapy in Rett, and then there will be an opportunity to expand out to more and more patients, assuming success in those early trials.
Yep, good point. Here's a good question. This is from Sumit. How do you make it specific to MECP2 mRNA?
This is a very good question. So the RNA Skip platform, those engineered tRNAs are really recognizing that triplet stop codon nucleotide. And so there is nothing that makes it specific to that nonsense mutation in Mecp2. But there is a lot of natural biology that these engineered tRNAs are not effective at a natural stop codon that falls at the end of an mRNA transcript. And there's a number of different reasons for that. One is the number of proteins that assemble to stop protein translation at the end really doesn't allow for these tRNAs to come in. Whereas at a nonsense mutation that is, say, in the middle of the mRNA transcript, you do not have that protein complex. And so those engineered tRNAs are able to slot in quite effectively. And so that's one of the major things that would enable it to have activity at that nonsense mutation and not at a normal chain terminating stop codon. So I hope that answers the question.
Another question here from Karen. Will the treatment be different for each case of mutated MECP2 or deletion, or is it generic? That's a good question. So does one therapeutic address all nonsense mutations?
So with a single drug product, we would be able to address all of the arginine to stop mutations. So the R168X, R270X, R294X, any others, even incredibly rare arginine to stop mutations, we would be able to address with a single drug product.
Here's an interesting question from Claudia, which I think I will answer. She says, I understand you can't talk about RNA switch, perhaps because it's proprietary. So I have to ask, why is this work being done by a private company instead of public research institutions? So almost all drug development is done in private companies. Basic science is done in academia, funded in large part by the NIH. And that's where discoveries get made, but that's not where drug development happens. And so drug development happens in private companies. So discoveries that are made by academic scientists, they file intellectual property, and then that intellectual property is licensed to companies. So it's a really important step that has to happen. And without private companies and investments, we really wouldn't have any drugs.
Yeah, and the one thing I'd add to that is I think even in my time in the biopharmaceutical industry and in academia, I've really seen a shift where you now have much closer partnerships between companies and academia. And so we very often use things called sponsored research agreements, where we are funding research in an academic lab and really establishing partnerships. Because I think, as Monica stated, there are certain things that, the raw pursuit of knowledge in academia enables a lot more freedom and perhaps more risk taking on very, very, very early stuff that a company would fund, but perhaps not do ourselves, because we really want to focus on how we translate those discoveries into the clinic. And so I think I've really observed, particularly over the last decade, that you have this really close partnership between biotech companies and academia that is really fueling the advancement of these novel technologies towards the clinic in a much faster way. And so I call it the kind of the ecosystem that everyone plays a critical role.
Here's a question from Yelena. Is there a risk associated with the AAV delivery system of mutating and becoming viral itself after it's been administered, or triggering the body to attack itself and rolling out an immune response that could possibly be detrimental?
So a number of good questions that are kind of all mixed in there. So what is done with natural AAV requires quote unquote help to be able to replicate. And so as part of what is done when using it as a delivery vehicle for a therapeutic is you take out all of the components that are not critical for its ability to infect a cell, but you do not keep in anything that allows that to self-replicate. Another thing about AAV is that unlike some other viruses, it has a very low to non-existent integration into the host genome. And so when we talk about that piece of episomal DNA, it sits outside of the normal chromosomal DNA in a nucleus. And so that also has a safety advantage because you would not be disrupting any natural site if it was to integrate into the host DNA. And then the third part of that, are we concerned about any type of consequential immune response over time? What I think about is that AAV itself has been shown that it does not cause any type of widespread immunity to the capsid once it has infected a cell, because that becomes degraded. The concern is that your payload that you're delivering, if the body is seeing that as foreign because that is consistently expressed in the cell, that is the real challenge and the real risk of an immune response. And I think that comes back to the fundamental view at Shape, where we want our payloads to be natural components that are present in the cell. And even our engineered tRNAs, the way that we engineer them, it still looks to the cell like it's a normal tRNA that can just slot into that protein translation machinery.
Great. Here's another question. Is there a chance for the RNA skip technology to skip the wrong nucleotide and cause another type of mutation different from the original one?
So there is not, because if you think about it, really what it's doing is it is competing with other tRNAs that as they slot in, it's basically looking for, do you have a match? I think of it almost as Legos sometimes, but many diverse linking systems. And so we're putting in a tRNA that will only link to a specific 3 nucleotides that it's reading. And if those 3 are not present, then you don't have a ability to bind into that ribosomal pocket and to place the amino acid there. And in essence, we are not competing with other tRNAs because there is not a tRNA that encodes for those nonsense mutations.
Okay. I think we already answered this question from Matthew, but I just want to be sure. He says, are we talking about personalized medicine specific to Rett syndrome or personalized medicine specific for the Rett patient?
So that is a good question. So for the RNA skip technology, one of the beauties is that it is able to be applied across all people with the same type of mutation, and that would be these arginine to opal nonsense or arginine to stop codon mutations. So with a single drug product, we could cover that 30% of patients in Rett syndrome. One of the other things that I didn't mention is that there are many other rare neurological or neurodevelopmental diseases that have some frequency of an arginine to a nonsense to a stop mutation. And if you look across all disease-causing nonsense mutations, around 20% of them are because of a mutation in arginine to a nonsense mutation or to a nonsense codon. And so this is a treatment that, in theory, you could apply it outside of Rett syndrome to other types of neurodevelopmental indications. And so that is something that we are thinking quite hard about. Also thinking about how that influences not only our preclinical research, but also how we would consider conducting a clinical trial.
Okay, and I'm going to take one more question. This is from somebody outside the US. I'm sure we have a lot of people outside the US on the call, on the webinar. And the question is, will clinical trials be accessible for people in other countries? So I'll just answer this. When a clinical trial has FDA approval and the protocol is made public, the clinics that are participating are listed with their contact information. And anyone from the US or outside of the US can contact those clinics to get more information or to schedule a visit to learn more and see whether your child might be a participant in the trial. So that applies to anyone. And the other question I get a lot is, what happens if the FDA approves a therapeutic that is a genetic solution to MECP2? Does there need to be trials in other parts of the world? And I think in general the question is probably not. We're not doing drug trials here. These are trials that are typically going to be quite small, very expensive to run, that are targeted at the genetic root and genetic root of the disease. And it's likely that the agencies that are like the FDA, but in other parts of the world, would take the data from a trial that happened in the US or in Europe, for example. So it's unlikely that companies would need to run trials in Europe and in Asia and Africa and different parts of the world. So hopefully, the data that would be generated from the original trial, which are typically done in either Europe or the US, would suffice. So that's a general question, but companies will have to interact with the various government agencies in different parts of the country, different parts of the world, when that time comes. Do you have anything you want to add to that?
The only thing that I would add is I think, the goal for a lot of people that are targeting genetic medicines is that you wouldn't have to run a multi-thousand person study where you're trying to look at small statistical differences to determine whether the therapy worked. Really, the promise and the hope of these truly genetically targeted medicines is that the benefit would be so obvious that it would make it then that much easier to see that benefit. And then it is a much stronger rationale for ease of not only expanding the type of patient that can be treated as far as age ranges and things like that, but that you would also be able to make it more widely available that much faster.
Yeah, that's a very good point. Small trials typically mean that the sponsors of the trial expect to see big changes that can be seen in small numbers of patients. When you see big trials, that typically means that the change that you're expecting to see is so small that you need to statistically power the clinical trials so that you can see that change. And that means having lots of people. So that's a good point. Well, I want to thank you so much for joining us, and I want to thank the families for asking such great questions. Happy holidays to everyone. 2020 was a difficult year. I hope certainly that 2021 is better for all of us. Be well, stay safe, and happy holidays. Thank you.
Thank you, Monica.